Hi, I'm Dr. Elizabeth Atkinson. I'm a statistical genomicist. My research is focused on improving methods to work more effectively across diverse human populations. Today I'm going to talk about 4 different DNA sequencing technologies, really get into how they actually work, and do a little bit of comparing and contrasting of the different techniques, specifically considering project design and which method might be best depending on your kind of study goals. This will be sort of human genetics focused, but definitely the techniques are applicable across other genomics contexts. All right, so I'm going to start off talking about Sanger sequencing, and we're going to take a kind of roughly historical order in going through these DNA sequencing techniques. So starting off with Sanger sequencing, the sort of implicit understanding in how all of these DNA sequencing technologies work relies on understanding complementary base pairing. So literally we can exploit the principle of complementary base pairing to use to actually get an understanding of what the DNA sequence is. So the key intuition behind why DNA sequencing technologies work is really just the understanding of complementary base pairing. So we know, for example, that A and T pair together, C and G pair together, and we can exploit this knowledge to figure out the DNA sequence on a strand using a lot of the same molecular machinery that our cells actually use. So the first technique that did DNA sequencing was called Sanger sequencing, and we're going to get into exactly how this works. So here's a sort of overview of the technique of Sanger sequencing. We're going to go through each of these bits in a lot of detail, but this is sort of the bird's eye view perspective of how we get from a DNA fragment to actual nucleotide calls. So we start off with our DNA template strand. This is a basically single-stranded piece of DNA that is what you actually want to sequence. So this is after you've done your wet lab work, you'll end up with these single-stranded bits of DNA ready for sequencing. In terms of the other ingredients needed for Sanger sequencing, you might recognize a few of these if some of you have actually done Sanger sequencing before. And also DNA polymerase is the same enzyme that our cells use to replicate DNA naturally. So the other ingredients include primer DNA. So these are short strand of DNA that are complementary to a piece of the template strand. So you'll notice that this TGTT is the same as the bottom of this template strand here, the ACAA. So we know that this primer piece will attach to this location on the template strand. We also have to throw in some standard nucleotides. These are the DNTPs in blue. So these are your traditional A, T, C, and Gs, as well as versions of your nucleotides that have different colored fluorescent tags attached to them. So the tags are specific to which nucleotide is there. There'll be a different color for A, T, C, and G. It's also important to note that these fluorescently tagged nucleotides lack the hydroxyl group. This is what actually allows the DNA nucleotides to continue kind of being extended in the sequence. So when you attach one of these fluorescently tagged nucleotides, you terminate the sequence, and you can no longer add any more additional nucleotides after that one is incorporated. So Sanger sequencing is sometimes called the chain termination method, and this is why it kind of gets that name. Okay, so getting into how Sanger sequencing actually works. As I mentioned before, this primer sequence will anneal to its complementary portion of your DNA template strand at the bottom of the slide here. And then DNA polymerase will randomly grab nucleotides from the pool that's floating around in your solution. So in this example, the first nucleotide that it would grab is complementary to the G over here, so it will be a C. It'll continue grabbing nucleotides until it eventually ends up grabbing one that has a fluorescent tag, which, as I said before, will end the elongation of this DNA strand. It'll terminate the chain. So you end up with a fragment of some length, which varies, the end of which has a fluorescently labeled nucleotide on it. So here's this sort of shown in practice. In this first example, you'll see that the very first nucleotide grabbed was actually fluorescently labeled. So that's as long as this chain ever got, just one nucleotide long. And you'll end up with a bunch of different strands of varying lengths depending on how many cycles of nucleotide addition were completed before hitting a fluorescently labeled one. All right, so getting into how we're actually sequencing the nucleotides, this works very similarly to your standard gel electrophoresis. The kind of primary difference is that now we have this long kind of capillary filled with this gel matrix instead of kind of a square plate. But the actual process of separating strands by length works very similarly to what you might have done in a lab. So the shorter strands are going to pass through this gel matrix more quickly than the longer strands. It's going to take them a longer period of time to navigate through the molecules. There's also a laser watching as these bands pass through your capillary. So when a particular fragment hits the laser, the fluorescently tagged nucleotide at the end of that fragment will emit its characteristic burst of light. So the light will be different depending on what fluorescent tag is there, which is in turn dependent on what nucleotide is actually present. So this is how you can assess what nucleotide is at that position. And because we know that, as we saw before, the sort of length of the strand tells us how long that fragment is and therefore what position in the DNA sequence that nucleotide is, by sorting the strands by length, we can get the order of bases. So the shortest nucleotides are going to be sequenced first. Those are kind of earlier in the sequence. The longest piece is last, and those are the end of the chain. So what you actually get from the machine is this sort of wavelength of light emission. So you get the kind of wavelength associated with the fluorescent tag that in turn is associated with that nucleotide. So it's kind of an indirect way to actually assess what nucleotide is there. But this is the core way that all DNA sequencing technologies that we're going to be talking about today actually assess the nucleotide. It's by interpreting what wavelength of light the machine thinks that it perceived at a given spot. All right, so here's an example of some very good output from a Sanger sequencing machine. You can see these characteristic peaks that are colored according to the perceived color of the machine there. And you can note that it'd be pretty easy for the machine to call which nucleotide is which. They're very discrete, they have a very clear order, they're all pretty strong peaks. You can be pretty confident that each nucleotide at a given spot is what it is. Of course, life isn't always perfect. Here's an example of some extremely imperfect output. So this would probably be data you would not be able to use, but I wanted to show this example just to give you some sense of how tricky it can be to call bases sometimes. You can see how difficult it would be to differentiate which base is present at any given spot, in addition to deciding sort of the order of the nucleotides called here. So this can happen from things like DNA contamination or just really poor quality DNA to begin with. So when you do your sequencing, the machine will output a quality score that tells you basically a metric of how confident you can be about the call at a given position. These are called Phred scores, P-H-R-E-D, and a Phred score of 20 is generally considered to be of good quality. So that's about a 1 in 100 chance that you get the wrong nucleotide called from the machine. Sort of a theme of the rest of this talk will be considerations for downstream filtering. And one of the primary considerations for Sanger sequencing is taking into account the Phred score, how confident you can be about the quality of the nucleotide call. So we've gone over the basics of how Sanger sequencing is actually done. This is still used to validate sites, even though there are sort of more modern techniques that are used. Sanger sequencing still is employed. It has the lowest error rate of the various sequencing technologies. So this is kind of why it's still of utility to research. So now we've gone over how Sanger sequencing actually works. The sort of key point that's shared with other DNA sequencing technologies is this idea of using fluorescent tagging to actually decide what base is there. So perceiving, calling what base is present based on a wavelength of light. So that's the underlying method by which all of these sequencing technologies actually decide on what bases are present.